Hello, welcome back to another video. Geez, it's been a while since I've shown a project. You know what? Every living thing on Earth is built from cells, but not all cells are built the same way. Biologists divide all cellular life into two fundamental categories, eukaryotic and prokaryotic. And the difference between them is really one of the biggest dividing lines in all of biology. The core distinction comes down to this. Eukaryotic cells have a true membrane-bound nucleus that houses their DNA, along with a whole system of other membrane-bound organelles, compartments that each handle a specialized job. Prokaryotic cells have neither. Their DNA sits directly in the cytoplasm in a region called the nucleoid with no membrane enclosing it at all, and they have no internal membrane-bound organelles whatsoever. In fact, that's literally where the names come from. Eukaryotic means true nucleus, and prokaryotic means before nucleus. Eukaryotic cells are typically much larger, often 10 times the size of a prokaryotic cell, and they include everything from animal and plant cells to fungi. It's a fun word to say, fungi, sorry. Anyways, prokaryotic cells, on the other hand, are bacteria and archaea. I never say that right. Remember, I had a biology teacher that was always correcting me on how to say certain words. Anyways, they're actually the oldest, archaea, they're actually the oldest and most abundant form of life on the planet, with a fossil record stretching back over 3 billion years-ish, according to academics, right? This is what they teach us. So with that foundation, let me walk you through what this project actually explores, the specific organelles inside each of these cells and what each one does. And that's what we're about to look at. So a quick look at Visual Studio Code. There's quite a bit going on over here. I quite literally just got done with this. I spent about a good 2 hours debugging and pulling my hair out of my head, not literally, but I had a lot go wrong with this. Okay, so I'm going to go ahead and spin this up, and then I will pull it over from my other monitor. Change directory into the client, npm run dev. All right, so I'm going to go ahead and cd into the server. Same thing, npm run dev. Okay, now over here, I'm going to, okay, let me pull this over for you. And here it is. Okay, I decided to go with this whole wireframe approach. Me personally, I think it looks cool. Is it 100%? I would say almost. This is the vision I had in mind. This is what I wanted to go for. Inside, let's take a look. So we're currently looking at an animal cell. Inside a typical animal cell, the nucleus acts as the control center, protecting the cell's DNA and regulating which genes get expressed. The mitochondria are the cell's power plants. They convert nutrients into ATP, also known as adenosine triphosphate, the energy currency every other process in the cell depends on. Then you have ribosomes. Ribosomes are the protein building machinery reading genetic instructions and assembling amino acids into functional proteins. And remarkably, ribosomes are found in every form of cellular life, eukaryotic and prokaryotic alike. The endoplasmic reticulum comes in two types. We're going to take a look at some of these organelles here in a minute. Your rough ER, studded with ribosomes, which builds and folds proteins, and smooth ER, which synthesizes lipids and detoxifies harmful substances. From there, the Golgi apparatus, I love that, Golgi apparatus, that acts like a packaging and shipping center, modifying and routing those proteins to their correct destination. Then you have lysosomes. They handle waste breakdown and recycling, and vesicle shuttle materials between all of these organelles surrounding it all. The cell membrane controls exactly what enters and exits the cell. I could talk a little bit more, but let's go ahead and take a look at this. What do we got here? So I decided to, at the very bottom, I integrated kind of a quick guide as the difference between eukaryotic and prokaryotic. Now I'm not going to read all of this. This took a while to type out. So here's all this. I had a lot of fun with this. I've been wanting to do something like this for a while because I love biology. And, I was pulling out old textbooks from college. I was looking through a bunch of my notes. Yes, I keep all that stuff. And anyways, so in here, you've got eukaryotic versus prokaryotic cells. And I broke it down. You've got genetic material. So on the left, you've got your eukaryotic, and on the right, you have your prokaryotic. So genetic material, internal organelles, the size, ribosomes, cell division. You've got energy production. You've got cell wall movement, origin, and evolutionary age. And then at the bottom here, I've got a little summary here, which is kind of cool. All right, so here's this. I can move this around, right? And I can zoom out. Oops, I accidentally clicked. Let's go inside of the cell. All right, we're inside. So I can click on anything. Let's click on this bad boy right here. Peroxisome, a small membrane-bound organelle that breaks down fatty acids and neutralizes toxic byproducts. So you get a quick little summary at the top, but then if I read the full explanation, right, then I can read. I'm not going to read all this. Now I am using an API down here, so I can ask a question about the peroxisome. I could simply just say, what is this? And then I've got some images here, which is kind of cool. Let's check it out, what we've got. Peroxisome, so you can learn a little bit more here if you like. And then click here. Ooh, that's kind of cool. All right, now let's click on another organelle. Let's click on this bad boy right here, the mitochondrion. And so a brief summary, you have the organelle responsible for producing most of the cell's usable energy in the form of adenosine triphosphate, or ATP. You can read a full explanation, and then you can take a look at some images. Oh, that's cool. Electronic micro, oh my gosh, it's been a long night. It's a picture of a muscle. Let's take a look at another one. These are really neat. I remember a few years back, I went to a certain university and I got to take a look at an electron microscope. That was a lot of fun. I want to take a look at this one here. Oh, wow. That's cool. That's really cool. All right, let's click on a different organelle here and we'll move on to the other cells. Let's click on this one. I did not mean to do that. We're having a freak out here. That was my mouse. I apologize. Let's click on this, the endoplasmic reticulum, a vast folded membrane network that manufactures proteins and lipids, existing in two distinct forms, rough and smooth. I already talked about this a little bit, each with a different job. Again, I can click down and read more in detail. I can see some imagery. Oh, wow, that's cool. Endoplasmic reticulum. That is so cool. I want to see this one. There you've got your nucleus. There's the rough endoplasmic reticulum, and then there's the smooth endoplasmic reticulum. That's a pretty nice little image there. I like it. There's your Golgi apparatus, lysosomes. That's cool. I like that. All right, all right. And then you can also take a quiz, which I'll do. I'll take a quiz here momentarily, but for now, I just wanted to take a look at some of these organelles. I did it again and get back inside of the cell here. We'll click on this right here. This is the Golgi apparatus. Let's see what we got as far as imagery. Oh, I like that. That's pretty cool. Okay, let's take a look at the, oh, that's really cool. Dang. Okay. So this is actually from the real imagery here that you see in the project. That's actually being pulled from, well, you can see here, it's Wikimedia. But what's cool about it though is there's no, like, it's completely free. There's no actual API key that I had to throw into my ENV file. So not that it's a big deal to put an API key in there. I just thought it was kind of convenient. So anyways, all right, let's click on this. This, of course, is, well, the nucleus. Got some imagery here. Diagram, human cell nucleus. Okay, interesting. What else I want to click on this one here. That's pretty cool. Like kind of hand drawn. I'm digging that. Okay. All right, let's get out of this. Again, I can move this around, except maybe I shouldn't click on it though. I can click, move it around. All right, let's head on over to plant cells. So plant cells share most of these same structures that I was, talking about earlier and what we were just looking at. But with some critical additions, they have a rigid cell wall made of cellulose, giving them structural strength. Animal cells don't have, right? So they contain chloroplasts, which capture sunlight and convert it into chemical energy through photosynthesis. Something animal cells simply cannot do. And instead of many small vacuoles, plant cells typically have one large central vacuole that can take up the majority of the cell's volume, generating internal pressure, excuse me, that keeps the plant rigid and upright. Same thing here. I can turn, rotate, move it around. It's pretty basic. I know. I get it. It's a little cramped in here, but we're gonna go ahead and go inside. Let's click on that. Oh, nope, I did not. Let's get over there here. Nope. It's really cramped in here. This was actually one of the last things I was working on. Maybe I should have stuck to it, but it's okay. It's manageable. Wait until you see the prokaryotic cell. Wait till you see my flagellum. It's ridiculous, but I did the best I could. All right. We got to see some images. Oh, that is so cool. See, I'm nerding out right now. I don't care. That is so cool. I miss seeing images like this under the microscope. Wow. Okay. What else do we got? Was okay. Okay. That's kind of cool. So you can see, let me click on, okay, you know what? This image right here is clearly my favorite. So you can see why I made the cell look the way that I did. Let's get out of here. We're going through organelles here. So let's go over this really quick. I get ready to laugh. I think this is kind of ridiculous. Okay, all right. So that's right. Then there's the prokaryotic cell with my ridiculous flagellum. It's structurally it is much simpler, but no less remarkable. No less remarkable, right? Instead of a nucleus, its DNA is concentrated in the nucleoid region, unprotected by any membrane. So many prokaryotes also have a cell wall, but or what am I saying? Built not from cellulose like plants, right? But from a completely different molecule called I can see it in my head, but peptidoglycan. Is that right? Peptidoglycan? I think that's right. So some have a flagellum, as you can see spinning around back there. Um, And unlike the whip-like flagellum on certain eukaryotic cells, a bacterial flagellum is a genuine rotary motor that spins like a propeller. It's so cool. Many also have pili or pili. I can never say that right. What those are short appendages used for attaching to surfaces or even transferring genetic material directly between cells. And some bacteria are wrapped in a protective outer capsule, which helps them evade a host immune system entirely. And if you look at this, you can kind of see how he did a separation here. Oh, capsule. Okay, let me get back out. I apologize for that. So you can see, there we go. Anyways, look at that ridiculous flagellum. I'm gonna try to get inside this. This one doesn't behave very well for me. So bear with me here as I navigate the inside of this prokaryotic cell here. Let's start over. Let's go back here. Aim it about like so. Ah, I did it again. Aha. Nope. Bear with me here. We got to get inside the cell. Let me try this again. I had a rough time with this. Okay. Bam. Okay, I got it. There's the flagellum. I want to take a look at the images that were provided. Ooh, okay, this is cool. Oh no, okay. I thought it wasn't going to work there for a moment. I cannot, respectfully, I cannot read that. So let me try this image. Same thing. Cool images though. Okay, so maybe there's like, okay, that's kind of cool. Different languages. I'm digging it. Wow, okay. Anyway, this right here is wild how it's all set up, but I'm sorry, I couldn't figure out how to do it in this when I was doing the code. To be very honest with you, I have no idea. Um, I have no idea. But I wonder, now that I'm staring at this, I'm wondering if there's a way to get a bit more um, creative with how the flagellum is actually attached. I don't know. I know I was going kind of basic, and oh my gosh, I was trying so many different things, and sadly, that was the best I could come up with. Yeah, so whatever. Anyways, let's back out of this. I was just writing a note. So here is this. Oh, you know what? Let's let's go in the animal cell where there's a little bit more room. Let's click on this here. Okay, we already looked at images of the endoplasmic reticulum. However, you know what we didn't do is we didn't take a quiz. So, let's see. What gives the rough endoplasmic reticulum its rough appearance? That would be the attached ribosomes on its outer surface. So you hit submit answer. You're not going to know how you did until after you did it, but it does give you a description here at the bottom, which I originally did not have, but I decided to go ahead and do it. All right, let's go to the next question. What is a primary function of the smooth endoplasmic reticulum? Producing adenosine triphosphate, synthesizing lipids, and detoxifying harmful substances. I feel like I literally just talked about this. Why can't prokaryotic cells process proteins with the same complexity as eukaryotic cells? I would say it's because right here, they lack an endoplasmic reticulum to fold, modify, and sort proteins after synthesis. So let's go ahead and click on that, submit. Okay, now let's see results. Come on, baby, let's go. 100%. There we go. All right, whatever. Anyways, so I can show less, show more. That's pretty much all it is. I was going to integrate some sound effects, and the last thing I was going to do, but I honestly, I don't know how to do it. I'm gonna have to learn. I wanted to have like a full, um, like a full 3D tour, and I'm not even really sure how I would integrate that. So I'm like, you know what? Let's just have these cells slowly move on their own, kind of like, well, you can see them slowly moving on their own. Now I'm trying to think if there's anything else I'm missing. I don't think so. I think that's pretty much it. I have to say that the prokaryotic cell is my favorite just because of how ridiculous it is. I am really proud of how I was able to do the separation here, as you can see in the wireframing. I had a really difficult time with that. I was able to figure it out. This was hurting my brain. So one last thing I wanted to talk about this channel that I recently um, started is I'm doing this new playlist on this channel called, and I have a funny story for you, and then I'll turn this off. It's a playlist called Stories. It's something I'm not going to do all the time, but I really enjoy writing. I love to just grab my pad of paper right here, grab a pen or a pencil and just write. I absolutely love it. Well, the story I've got for you is I was a guy I work with, we were talking about it, and I was telling him, about my idea to integrate the stories playlist on my channel. And he said, you know what? Why don't you just have AI do it? And I thought, dude, let me just say this. If I woke up tomorrow morning and I found out that AI was no longer a thing, data centers are getting torn down, no more AI, people. AI is gone. Brother, I would celebrate and get hammered, right? I told my coworker, I'm like, no, dude. Like we live in a time where people are bragging about AI in such a way that it just baffles me. Oh, I can have AI check my emails. I can have AI. Yeah, do it yourself. Do it yourself, man. And like I've said before, I'm sure I would probably be able to pump out projects faster. I don't know. Call me old school, man. I mean, yeah, I use AI for the thumbnails on this channel. I use it occasionally, well, often for helping to debug or if I'm just absolutely stumped. But I'm stubborn, man. I look, you Google something and the first thing that pops up is an AI response. It's like, come on, man. Like fast, quick, convenient. I get it, but I feel like I don't know. I watched the movie Idiocracy not that long ago again, and it made me think a lot of AI. I'm just gonna leave it at that. So it takes me a little while. But anyways, stories. So um, I've only done one so far. I know there's a lot of channels out there that do them, and I just thought, why not? Why not? Um, you know, embrace my love for writing and do a stories playlist. But anyways, what was the last thing I was gonna do? Oh, I wanted to read this at the bottom. It says, despite these structural differences, eukaryotic and prokaryotic cells share deep similarities that reflect a shared revolutionary origin. Both are enclosed by a phospholipid bilayer membrane. Both use DNA as genetic material and the same universal genetic code, and both rely on ribosomes to translate that code into proteins. These shared fundamentals are part of why the endosymbiotic, geez, now that the word's in front of me, I can't even say it, endosymbiotic theory, the idea that mitochondria and chloroplasts descend from ancient free-living prokaryotes, is so widely accepted. It explains how one cellular strategy could give rise to the other. I'm not going to read all this. I've already read more than I should on this project, but there it is. There it is. That is my uh, that is my cell biology project, and I hope you enjoyed it. And this went way longer than I wanted it to, but I will see you in the next one. Thank you for watching and take care. Bye.